HRN listeners. As we celebrate our 15th year, we are deepening our commitment to giving voice to the next generation of food systems storytellers, and we need your help. Our internship and fellowship programs help activate new possibilities for underrepresented and underestimated young people through experiential journalism, audio engineering, and production training. Through these unique programs, HRN helps food equity stewards build essential workforce readiness skills that expand their potential and foster economic mobility. Please consider supporting these critical programs. And with a minimum donation, you can be entered to win a dinner for two at an amazing restaurant in one of 8 cities and tickets to a concert at a great venue in one of those cities. We have incredible partners across the country who have donated, as they also share our passion for helping to educate the next generation of food systems storytellers. Check out heritageradionetwork.org/15 to donate and enter to win today. That's heritageradionetwork.org/15 to donate and enter to win today. And make sure you donate before March 31st. Thank you.
Welcome to Heritage Radio Network on Tour. I'm Katie Mosman-Wadler, and today we are broadcasting from Day 2 of On the Rise, the International Symposium on Bread at Johnson & Wales University in Charlotte, North Carolina. Today's coverage is brought to you by Charlotte's Got a Lot and supported in part by the Julia Child Foundation for Gastronomy and the Culinary Arts. I'm very excited to welcome our next guest, Dr. Erin McKenny. She's a microbial ecologist, and she was the keynote speaker for On the Rise. Welcome, Erin.
Thanks so much. Pleasure to be here.
So what does it mean to be a microbial ecologist?
Oh, it means a lot of things to a lot of different people. But in my life, it means I'm intensely interested about the complex communities that live in your gut and in foods, and why different types of microbes live in certain places and not in others, and how they live there.
Awesome. And can you give us your background and sort of how you got interested in this field and your educational background so far?
Yeah, it's been kind of a long and winding road. So from age 4 to 24, like going way back, I wanted to be a veterinarian because I always loved animals. And that clearly didn't work out. But instead of getting to go to vet school, I did get to go live in Orlando and work at Disney's Animal Kingdom as a nutrition intern. So studying wild animals that we keep in captivity. How do you feed animals that live in the wild foods that are, commercially available, that humans have readily available, and still satisfy their nutritional requirements? So I got really interested in the ways that we can take care of animals best, and in nutrition as a foundation for good health. That's also where I learned about gut microbes, these hundreds of trillions of superhero sidekicks that live in your colon from birth to death, and they're helping you every step of the way. They're with you every day of your life, digesting your food, teaming up with your immune system to help you, fend away pathogens. They do incredible things. So for 8 years, as in a master's program at NC State University, and then as a PhD student at Duke University, I was studying gut microbes in primates and in these rogue carnivores that, originally might have eaten meat, but since then they've gravitated to all fruit diets, like the binturong, which is a rainforest dweller in Borneo.
What sort of animal should I be picturing right now?
They're also called bear cats. So picture kind of an otter-like face, long whiskers, but these like peaky little perky ears on top, like a cat. But then the thick, coarse black fur of a bear, but a really long prehensile tail.
Whoa.
They're wild.
Whoa.
Yeah.
Can they hang from them?
I have not seen that. I think they generally use them for balance.
Okay.
Yeah, I haven't personally seen them hang, but maybe they can.
Sorry, we got a little sidetracked there, but I'm fascinated.
Exactly. Binturongs, they're worth the side trip, right?
Yeah.
It's like the world's largest frying pan. Yes. So going from, all of these incredible animals and the microbes that live in their guts that help them digest different diets in different parts of the world, I was also teaching the public about gut microbes and why they're so important. So many people across all ages of life, they think when you say bacteria, it's got to be germs. And that is completely untrue. Most of the microbes that live on and inside us, like they cover us, they outnumber us. And if they were all pathogens, we would not be here because bacteria are really good at what they do, right? So most of these microbes are on our side or just along for the ride. So I was teaching, that sort of a positive message about microbes from kindergarten up through 12th grade, and in, public places like museums to students of all ages. And that got me on Rob Dunn's radar as a person who might be a good fit for the sourdough project, in which we're engaging the public to think about the microbes that live in sourdough starters from around the world.
So before we get to sourdough, I want to do a little more detail about your research of gut microbiomes. So as an animal nutrition intern, you're studying inputs, but also with your interest in gut microbiota, you are studying outputs as well. So can you talk about that project?
Yeah, that's a great euphemisation. So
we're going to start with the euphemism, but we're going to leave it behind very abruptly, I think.
Leave it behind. I see what you did there. There's so many puns in this work, right? Yeah, so working with wild animals, even in captivity, most of the species that I've studied are endangered. And lemurs in particular are the most endangered group of mammals on the entire planet. So you can imagine if you want to study the gut, you are not going to desire to or get permission to, put an animal under anesthesia so that you can cut open its bowels, right? So we want non-invasive ways that are not stressful to study these animals' insides. And what better way to study the gut than to study poop? Right? I mean, food, poop, they both have two O's, one's in, one's out. But also these fecal samples are being offered to us and provided to us with no stress by the animal, right? We hope, right? A healthy animal shouldn't be stressed if it's pooping.
Right.
But also, so it's a non-invasive way to get a look inside the bodies of these creatures. And it's happening all the time, right? Sometimes on very specific schedules. But yeah, so my glamorous life in grad school, I think in my bio, it usually says in grad school, I collected over 140 samples of poop from animals belonging to 15 different species at 4 different zoos, with other side projects. I think at this point it's way over 250 samples. So yeah, lots and lots of poop.
And what does that analysis look like? So you have your samples, you are looking at what are the organisms that are actually present there. What else are you looking for as far as like composition and number and comparisons, and then the kind of data science on the other end of that?
Yeah, so you're definitely on the right track. The first thing that I do when I get a fecal sample is put it on ice and then get it to a minus 80 Celsius freezer, like super ultra cold, as quickly as I can, because that stops the microbes from reproducing anymore. They're really quite happy in gut content. As you can imagine, they're surrounded by food. And to get the most accurate snapshot of life as it is in the gut at the moment that the animal pooped, you want to freeze down those microorganisms and stop them, freeze them in time, right? Otherwise, you'll get kind of a shift in the community structure. So we freeze them immediately. Then I take them back to the lab. I use a razor blade to shave away the outside of the poop because that has been contaminated by the environment, right? I have a colleague who can actually hold a test tube up to the bottom of an animal, and she can get lemurs to poop straight into her tube, so there's no environmental contamination. But
it's a special connection.
Yeah, I am not a poop ninja like Lydia Green. So, so my collection technique involves, fresh pairs of gloves and sterile tongue depressors to collect these things from the floor. So I shave away the outsides of these pellets. Then from the inside, that is, just a pure and true picture of what's happening in the gut, I'm extracting the DNA. So you basically kind of pulverizing that poop to make a homogeneous slurry, and then you're filtering out all of the food particles, all of the RNA, all of the proteins, any other contaminants, so that you get pure DNA. And that's from bacteria, from yeast, from viruses, from anything, even from the animal's intestinal cells. So the next thing that we do is send it off to another lab that uses they use fingerprint gene markers, right? So it's like 200 base pairs of DNA, so 200 letters. And 200 might sound like a lot. It's not a lot compared to all of the DNA in the host chromosome, right? Or in the bacterial genome. So it's pretty fast and cheap to sequence, but there's enough information in those 200 bases so that we can tell different types of bacteria apart from each other. So essentially, I get a file that might be 2 gigs at the end of the sequencing run. It's going to contain 20 million reads. And those might be from up to 200 or 300 different samples. So then I have to, this is what we call big data, right? The age of big data. I could never look at all of that by eye, much less, start to analyze it or make sense of it for my human brain without the help of computers. So I have learned to write some computer code and scripts so that I can tell the computer, throw away those sequences. They're trash. They don't have enough information or there are too many errors. And then the good sequences that are left, we can actually compare to each other to find out how many different types of bacteria are there. Kind of like comparing last names in a phone book, right? People with the same last name are most likely related. That's the analogy for what we're doing with this data. And then once we know how many different types of bacteria there are in the entire data set, then we can divide out.
And you're getting relative composition,
yes,
too. From
exactly.
That.
We divide out, how many of each type of bacteria are found in each of the animals that I studied.
Mm-hmm.
Yeah.
And what happens if you want to go deeper onto like a per species level and really understand like the full array of what is the actual makeup of that microbiome?
Oh, for the bacteria? Mm-hmm. So generally, your first pass, you're going to get kind of at the genus level, right? But within each genus, there are so many different species, and some of those might be better for the animal, better partners, and some of those might be kind of, worse for the animal as partners or for health. Mm-hmm. And the same is true, by the way, all of these techniques we also use for the sourdough project, right?
This is why I'm asking you to go into so much detail about this. Yeah.
It sounds very fecal-centric, but it actually works for food too, mm-hmm. right? It works for microbes no matter where they're living. So if you want to know the species types, so at this point, there are some computer analyses that we can run that do tell us from that kind of last name reading, kind of the species level, and we would call those sequence variants, right? So any type of sequence that is different from each other in any way, they could be different species. But the other way is to actually isolate a microorganism living, which is much easier to do from a food sample than a poop sample, right? Because the conditions in your gut, there's not a whole lot of oxygen in there. So it's actually very difficult to grow those different bacterial types in a lab. Mm-hmm. Also, do you really want to be growing the microbes that live in your gut just on a benchtop, right? So there, it's difficult for a couple different reasons, like safety protocols, oxygen limitation, all of those things. Mm-hmm. But from food, it's really easy, especially from sourdough starters. We've been able to culture most of the bacteria and the yeast types that live in these starters. And once you have, so you can imagine a Petri plate. So it's a circular dish, maybe 4 inches in diameter, and you're pouring agar into it, which is, it's a gelatin with a lot of nutrients in it. So it's almost like you're making ramen broth jello, right? And it has all of the salts, sugars, proteins, fats that microbes would need to live. So when you take a little bit of sourdough starter, dilute it in sterile water, put a few drops of that sourdough juice onto your agar plate and spread it around evenly, you're also spreading out the bacterial and yeast cells on that plate. So wherever one cell lands by itself, it's going to chew up all of those lovely nutrients in the agar. It's going to divide and multiply until it makes a colony that you can see with your naked eye. Some of these colonies are only as big as the head of a pin. They're very small, punctiform colonies. And then some of them will actually take over the plate. They might be, depending how long you let the plate incubate, they might be nickel-sized, quarter-sized, $50 sized. Those are going to be your microbes that are growing super fast. They can take over, any situation very quickly. The weeds of the microbial world, right? So all that to say, once you have all of these different bacteria or yeast growing on your plate in a live culture, then you can separate them. You can isolate their DNA. And because it's one type of organism, you can actually sequence the entire kind of gene region that would tell you not just the last name, but almost like the first, middle, and last name of that organism. So that's how we can get to a species level.
And throughout your career, have you seen the cost of sequencing affect the research that you're able to do in a relatively short time? And then what is going to be next that's going to be so amazing about cheap sequencing technology?
Yeah. So that's a really excellent point. And sometimes I feel like the hipster gut microbiologist, because I was doing it a little bit before it was cool, or I had started doing it right as it became really cool, right? So my master's program, I was using mostly Sanger sequencing, which is the gold standard, very low error rates. So it's almost a perfect run, right? You're getting kind of a perfect translation of what the names of these creatures are. But it can be very time consuming. It can be pretty expensive, and you can't get a whole lot of data. So just for scale, my entire master's project was 1,400 sequences, right? Which might sound like a lot. It felt like a lot because I was analyzing by hand in those days, right? Back in the day, But while I was working on that data set, Illumina sequencing hit the market, right? And Illumina platforms, my first Illumina data set in my PhD had 20 million sequences for one data set. And that's for, a MiSeq run where you're just taking the last names. So that's where big data comes in. And those two data sets, 1,400 sequences and the 20 million reads, they cost about the same. So with these, we call them next generation sequencing because it's the next gen, it's like the newest, most exciting. You don't often get better, faster, and cheaper. It's usually pick two if you're lucky. But I really think with this next generation sequencing, we've gotten all three. And so when I started, my first Illumina sequencing run was probably $4,000 or $5,000. But the prices have continued to drop, not only because the techniques are improving, but also the chemistries are improving. So we're actually able to fit more samples on a single run. So where my first data set, I put, I think, 160 samples on an entire sequencing run. Now you can fit up to 300 or 400 samples on a run. So it's amazing. Not only is it getting cheaper to sequence the samples themselves, but you can fit more samples in a single effort. So it's getting, cheaper and more efficient and more effective.
Yeah. And it's only going to keep doing that, I think.
Yeah.
So the sort of world of we could all understand, I think, our own microbiome as well as the world around us, which might be sourdough or other foods, or it might be our environment, I think is at our fingertips. And I'm not sure we know what to do with that yet.
Exactly. Well, and this is just the technique called amplicon sequencing, where you're kind of amplifying the signal of just those last names of the bacteria in the community or the yeast in the community, right? Another sequencing technique is called shotgun metagenomic sequencing, where instead of targeting just that last name fingerprint gene region, you're actually chopping up all the DNA from all the organisms in the entire community at once. So that tells you somewhere in that mix, the kind of alphabet soup of the genome, is the last name of all of the organisms. But also you're essentially able to then read the resumes of the entire community at once to find out not just who's living there, but what they're capable of doing, all the skill sets that they might have. And that link from the form of a community, like who's living there, to the function, like what they're capable of doing, that link is hugely valuable for starting to think about really why does it matter if you have specific microbes in one environment and not in another? And how does that relate to what they might be able to do, especially in systems like food or the gut that are going to directly impact the well-being of humans and other animals?
Yeah, and correct me if I'm wrong, but I think the pace for understanding the composition of those environments is so much faster than actually understanding the sort of deeper meaning of what that implication is for the food or for our health. And I don't see that necessarily catching up in the same way because it's not dependent on like a sort of a narrower subset of technologies. I mean, imagine you're trying to figure out like, okay, with human health, what does the gut microbiome mean for us? And there are so many processes that are affected. I mean, we have a vague understanding that probably our gut bacteria are influencing our thoughts and our cravings, but we don't really know how that's working quite yet. And there's so many steps sort of in identifying that. And humans are a really difficult research organism because we can't control the sort of external conditions to any one test, right?
You're absolutely right on all counts there. So not only are these communities incredibly complex, but it is incredibly difficult to say, okay, humans, we want to study you, only eat this type of food for 2 weeks or for a month. And we're going to clock all of your behaviors. The only way you would have the control, without shadowing somebody. And Jane Goodall can tell anyone that the moment you start to observe a study subject, they will start to behave a little bit differently, right? In the 18...
It's like, I'm going to follow 2 feet behind you and watch everything you do, but don't do anything differently.
Just
okay,
act
Erin?
normal.
Yeah, just like
exactly.
Pretend I'm not here.
Exactly. I mean, we've all seen The Office, right? At some point, you break the fourth wall and you talk to the person who's following you around with a camera, and then, the spell is broken. It's not normal life anymore. Like, that you're under glass. Mm-hmm. Yeah, so it's incredibly difficult, and some might say for those reasons impossible to get a perfectly accurate viewpoint of how these gut microbiomes are tied up in our every moment, of our lives. So a lot of researchers have turned to model organisms, right? And you can get a great deal higher control with lab rats or mice, or even with captive primates, right? If you want kind of a step closer to the complexity of a human life. Mm-hmm. And in a more naturalized captive environment, right? They also, like humans, live in captivity, right? We don't think of it that way because we want to say, I love this house. But your house is a captive environment. You're not out in nature, in the world, really,
Right.
Yeah, so I think it's just really difficult to get a perfect view of that. And everything is so context dependent, as much as those mechanisms can be determined in those perfect model systems, how relevant are they to humans really? Mm-hmm. Yeah.
So let's talk about bread, because bread we have a great deal more control over, but it's also incredibly complex. So we spoke a little bit with Guylaine Lacaze about the sourdough project yesterday, but in case anybody missed that, can you give just a quick overview of what the sourdough project is and sort of the purpose of studying microbial environments within bread and fermented foods?
Yeah, so our global sourdough project in Rob Dunn's lab at North Carolina State University is an effort to try to sample and characterize the microbial diversity associated with, spontaneously fermented, we like to call it wild bred sourdough starters from around the world. So if you're looking for the maximum diversity that occurs in the world, you actually want the least amount of control, right? So instead of asking participants to grow their own starters from scratch using these set ingredients, we reached out to the general public with a survey 3 years ago and said, if you have a sourdough starter, please tell us about it. Tell us how old it is, where it came from originally, if you know who made it or what company you procured it from. Is your body or the body of whoever is maintaining that sourdough starter male or female? Do you have pets living in your home? What type of home do you live in? What type of flour do you feed your starter? How often do you feed it? What type of water do you use? I mean, we ran through, I think there were like 70 questions, just everything we could think of at the time that might predict or inform, or shape the communities living in these starters. Mm-hmm. So we sent that survey out to the public. Over 1,000 people responded. this is an incredible return rate, right?
Yeah, I mean, I did a survey for my graduate research. It was a narrower population than that, but that's a great number to work with.
Yeah.
You can really learn things from 1,000 samples.
Really incredible, right? So we got those 1,000 responses back, and at the very end of that survey, we'd said, oh, P.S., if you would like to know what microbes are living in your sourdough starter, then send us a sample of it. And over almost 571 people from 17 countries around the world sent us a cup of their starter in the mail, like triple bagged in Ziploc baggies, to Tufts University in Boston, where Ben Wolf's lab received them and processed them. And that's over 50% rate of response from 1,000 people. I mean, 50%.
It's unbelievable. Breadheads.
Especially, yeah, breadheads for sure.
Thank you, breadheads.
Yeah, it's like Rob Dunn's lab has a number of public science efforts that engage the public to learn more about the science of life around us. There are projects on cats. There are projects on pumpkins, on peppers, on chilies, on ants, on shower heads. I mean, usually if you get a 10% response rate, that's more typical. 30% is awesome. So that we got 50% of our survey respondents to send in samples. And there are people still occasionally sending us samples. And we're like, oh, we're really sorry. We finished sequencing like a year and a half ago, you know? But it's been incredible. And not only did they send us all of their samples, so they're deeply invested in learning about what's in their starters. A number of these participants joined the Sourdough Project group on Facebook. There are 350 people in that Facebook group, so which is a high percentage of the 571 that sent us their starters, right? And they're on there every day. There's a new post or three of, you know, I'm participant number 120, just off the top of my head, not a real post, you know. Here's the latest bread that I've baked with my starter. And they share recipes. They share, you know, hopes and dreams and failures. They ask questions. They're also engaging in the scientific process, which is something that we really hadn't had from another group of public science participants before.
Cool.
Yeah.
I mean, that's just amazing. And I think it really like speaks to the community of people who are like really, really caring about being hands-on about bread. So I want to know what you learned from the samples so far, and you know, to what extent can you learn about human culture from sourdough cultures?
Oh, I love that. Yeah, that parallel in the word culture is just like a driving force in my life right now. It's so humbling, right? And that love is all around us. So what we've learned is that no matter where you live across, when we compare the microbial communities of all the starters in the world that we've collected so far. And the caveat there is they are Western centric, right? So most of our samples came from the United States or countries in Europe or Australia, right? We didn't have a tremendous representation from other countries. So that's something to keep in mind, right? This may be the truth of a certain subset of the world's sourdough starters, right? What we find is that sourdough communities tend to be, for all that they're so stable, they tend to be quite simple. And for the most part, only between 1 and 4 or 5 different types of bacteria and yeast make up 99% of the community.
So explain for a second how that is possible, because you have this living culture that is being fed regularly, and if you imagine you're leaving food out, you know what happens to food that you leave out. So how can it be that you've got this living thing full of nutrition and great growing conditions, and you're not growing 10 million different species that are all competing with each other?
Right. So and that's a question that we asked ourselves too, like, why are we seeing, you know, it's when we compared all of the microbial communities across all the starters, we came up with this heat map with these beautiful banding patterns, right? So it was like building the family tree of sourdough starters. The more similar your microbial communities are, the more closely related they are on this family tree, right? And what we found when we ran that analysis, we call it a cluster diagram, is that there seemed to be 15 different clusters of different types of sourdough starters based on the microbes, the bacteria, and the yeast that are most dominant in them. So if you put a heat map on that, imagine just bands of red, and you see different banding patterns for each of these 15 types. And there are a couple families where you can see more rare types too, right? Mm-hmm. But for the most part, you just see these big blocks of red that show, well, this is type 1, this is type 2, this is type 11. So when we then asked, how can we explain these patterns in the membership that we're seeing, we found, we tried to correlate what the participants had told us in their surveys, all of those human-related things, variables, from how they manage and take care of their bread to how they live in their homes and where they live in the world. We found that, by and large, where you live in the world, geographically speaking, probably to do with climate, that tends to correlate most with the fungi, right? Now, yeast are a type of fungi, but we even found that this geographic signature tends to be left in the molds, which are very rare. Yeah, so it's like a very faint geographic fingerprint of where you live. And the bacteria follow a totally different rule. They don't care where you live, they care how you live. So no matter where you live in the world, if you feed your starter rye flour or wheat flour, that has a different impact on the number and type of bacteria that live in your starter. Or whether you keep your starter out on the countertop all the time or put it in the fridge for 3 weeks. My starters are cold and lonely right now while I'm here, right? That also has an impact on the bacteria. So we know that bacteria and yeast are following different rules, but all of those human factors combined only explain 10% of the variation.
So where does the rest come from?
So that's the question, right? And it takes humans down a peg because we love to think, I know how to make this bread. We're here this week surrounded by bread experts, right? Who can take a given starter, a given type of flour, with a certain amount of water and a certain temperature and amount of time. They can give you a defined end product that is consistent. And that has got to do with the human, but it is also driven by the microbes, and that's what we don't understand yet. So to get more to the heart of that question, how are the microbes contributing not only to the breads that we eat, but to the patterns of diversity that we're seeing in these sourdough starters? We turned back to those cultures at Tufts University. We took, and we asked, is it something about each of these microbes themselves? So when we grew up and measured the growth rate of those most dominant bacteria and yeast that we find in starters all over the world, we find that the ones that take over might be taking over because they grow so much more quickly than other bacteria and yeast. It's like backslopping is kind of like mowing your lawn, right? Every time you mow the lawn, you're cutting down the grass and some weeds, but you're also preventing trees from growing. Anything that grows more slowly is never going to be able to take over a lawn. Unless
you can't keep up.
Right. So when you backslop every day, you're removing like half of the community, right? It's like cutting the lawn of your sourdough. So you would be, chopping that tree back down to a sapling. It's never really going to grow, those slow growers. But the weeds do really well. So that's where Saccharomyces cerevisiae and Lactobacillus sanfranciscensis and the other top most dominant taxa, they tend to grow really quickly. So sourdough starters favor the weeds of the microbial world.
So I used to work with Saccharomyces, and I'm a big fan. But in the bread community, it's becoming a little uncool. Cerevisiae. So what's the story there? Because, it's something that humans have depended on for a long time. Why does Saccharomyces cerevisiae now have kind of a bad rap in the bread world?
So the story that I am uncovering, right, and because, again, I'm not a professional baker. I'm a professional microbial ecologist, right? But the story that I'm gathering from the bread community is this idea that, the commercial yeast, the commercially available yeast, which is Saccharomyces cerevisiae, that specific strain that's commercially available was, pun fully intended, bred in a lab.
Bread.
Yeah, exactly. So it was bred in a lab specifically for rapid rise properties. So it's really good at chewing up sugar and starch and converting that to carbon dioxide. Tons of carbon dioxide. That's all the bubbles in your bread that make it rise. So you get a rapid rise really fast because the commercially available Saccharomyces cerevisiae strain is so good at that. But that means if it's producing mostly or only carbon dioxide, then it is not producing any of the other volatile compounds that we would call, volatile organic compounds, or the aromas that contribute to the complex smells and flavors of bread. That's why a commercially produced loaf of bread, or a loaf of bread that you bake at home with commercial yeast, it's going to smell yeasty for sure because there's yeast in it.
Right.
And it's going to have a lot of air in it. But it's not going to smell or taste anywhere near as complex as a loaf of bread that you bake with a sourdough starter, right? So I think there's getting to be some kickback, culturally when these yeasts came about. And I always think for the U.S., like this probably came about in the era of fast food. You wanted to mass produce food that was all the same. There was a comfort in standardization. So there was, everyone really embraced these light, fluffy loaves of Wonder Bread that were all the same.
Yeah.
You have control. And I think where we're going now more culturally is a shift toward flavor. That we don't mind necessarily that it takes a bit more time or effort as long as that flavor is there. We want the real experience of the foods. And to get that, we've got to turn away from the commercially available Saccharomyces cerevisiae to all of these other strains and variants of Saccharomyces cerevisiae that are occurring all around us in breads across the world.
Mm-hmm. Awesome. So now that you guys are kind of advancing the sourdough research, what are going to be the sort of next steps there, and what might be takeaways for professional or really dedicated home bakers?
Yeah. So now that we feel like we kind of have a handle on who's living in these starters, and at least one of the aspects of why those starters might be so dominated by these very few but very specific types of bacteria and yeast, the next obvious question is, how do those different microbial communities affect the bread that we're baking from those starters? So first, we actually revived some of the starters in test tubes and fed them the same flour and water inputs. So we controlled their nutrition across the board so that it's all the same, so that the differences in the dough rise, which we filmed over 36 hours. And I say we, but this is really my collaborators in Ben Wolf's lab, Liz Landis at Tufts University. She set up all of these test tube racks, fed all of these sourdough starters the same flour and water, filmed them for 36 hours, and used computer software to measure like the pixel difference in how much sourdough was, rising and rising in each of those test tubes.
Wow.
So using those pixel differences, she was able to graph growth curves over time. And what we're seeing is it looks like those different clusters of different sourdough types do tend to show different rise properties. And then, of course, it's not just how high your bread gets, but how it smells and tastes. So the other big question we had was, how do those different communities, or do they produce different aromas? Mm-hmm. So to do that, we took this, the same set of starters that we grew for the dough rise experiments. We gave those starters, again, at 24 to 36 hours to a sensory evaluation team at Tufts University.
Cool.
Yeah.
Cool job.
Oh, they are incredible. I call them sourdough sommeliers, right? Because they are able to detect very specific scents. Very, we have recordings of these sessions, and it's like I could just listen to them forever, maybe, because they're passing around each of these starters, and they're like several rapid sniffs. And then, if you need a relief, this is a pro tip that they gave me. If you are ever smelling something and you need a break, like you're having some sensory fatigue, smell the back of your hand. And it's something about smelling yourself resets.
Takes you
yourself.
back.
Yeah. Like, who needs smelling salts? If you get the vapors, you just, smell the back of your hand and you'll just reset and be all right. Yeah, it's a nice like coping mechanism, so they're passing around these starters and you just hear them like, oh, fruity, floral, rose, or brown fruit, or there were some, they're like, oh, fecal, and and they sent us a list of these descriptors, the top modifiers that they used to describe a lot of the different sourdough starters, and they're the most commonly used descriptors were highlighted in red, and one of them was like burnt perm hair.
That's very specific.
Right. I don't think that I have a marker for that in my brain, right? But they, while I was visiting Boston last week, I got to meet with the sensory team, and they happened to be doing a different, flavor comparison. I think it was of milks, right? And so they said, oh, sip this. And I'm like, what is that? And they're like, rubber glove. And as soon as you have the words for it, you're like,
right.
That's absolutely rubber glove. How did I not know? How do I know what rubber glove tastes like? But like, somehow
it's in there,
right? Yeah. Oh, it's definitely in there. So rubber glove was not an aspect of the sourdough starters, but they trained their brains and their noses together to come to a consensus across these 3 specialists, and they had this list of this is what we smelled in these different starters. You can imagine we only had them do a small batch at a time because nasal fatigue is a real thing.
Yeah.
And it, at some point, even the back of the hand can't bring you back from that fatigue. So once they trained their brains, then we actually took a sample of the smells. We kind of used, it's called a twister bar. It's magnetic so that you can use a magnet on the outside of the test tube lid to suspend this sticky bar that collects all of those aromas from the air above the sourdough starter that's growing in the tube, right?
It sounds very magical looking.
It's awesome.
It sounds like a magic trick.
It looks like it's defying gravity. Yeah. Exactly. So
you just take your hovering smell collector
right
inside your tube of air.
As you do.
Yeah.
And then, after the requisite 12 hours of smell collection, then you put it into this tiny little glass vial and load it onto a gas chromatography mass spectrometer, or GC mass spec. And it is probably between 3 and 4 feet wide. It's between 2 and 3 feet tall, right? And you load this little smell magnet collector into the input, and it kind of bakes off all of those smells and gets them back into the air, and then collects them into this really long tube. And it's a really long tube so that all of those different compounds that are responsible for the smells separate off by size. So the lightest weight compounds, the smallest, tiniest ones, they zip down that tube really fast and they burn off first, right? Mm-hmm. So when they hit the flame, then a signal goes to the machine that reads, oh, there was this much. It writes a peak on this graph that it's building. So if there's a whole lot of that first compound, then the first thing, it's almost like a seismograph, right? But it's for the amount of these different compounds that weigh different amounts. So they come off at different points in this reading. So at the same time that it's being read, half of the smells have been split off to be quantified and identified there. Half of them go down another tube to what we call the ODP sniffer device. And it kind of, it's twisty at the base, almost like some of those CFL light bulbs. Mm-hmm. But then it ends in this flared little plastic funnel that you stick your nose all the way into. So then you've got these sourdough sommeliers who've already trained their brains and their noses on the starters. So then for each starter, as the smells get baked off and identified, they've got a headset on so they can dictate the smells. They've got their nose buried into this little plastic funnel, and they have their finger on this remote control to mark the intensity of, on a 4-point scale for like how strong the smell is.
So they're actually labeling the mass spec peaks in real time.
In real time. They're
that
dictating.
Is so cool.
Yeah. And then their dictations are annotated onto the graph for like, oh, for this peak, you said woody and roses. And then you have to go back after that and see how big was that peak? Was it a single compound, or was it probably many compounds, right? And the whole time they've got their nose buried in this funnel, they're just like, like circular breathing. It's, sometimes you need a break just to make sure that they don't hyperventilate, right? It's incredible. It's incredible.
This is amazing.
So that data is still coming in. But what I'm seeing for the future of sourdough starter research, to bring us back to your question, I feel like with all of these different big data sets that we've collected from this global public science project, I feel like we have an opportunity to really understand why specific microorganisms might be highly dominant in different sourdoughs. Like, why do we see these 15 different sourdough types and not 17 and not 5 different sourdough types, right? But it also, why do we see them regardless of where people are living in the world, or how they're treating their starters? But then also, if you have that microbial makeup, how those microbes might then be affecting the flavor and the rise characteristics of your bread. So you can imagine if somebody really wanted to apply all of this knowledge, I'm thinking of as a sometimes home brewer, right? We always think, oh, what do I want to make? And you walk into the homebrew supply store and, you get like a clone recipe, like, oh, if you want an Abbey double, then follow this recipe. And it's with a specific types of malts in a specific ratios, specific yeast strains, specific hop varieties. Can you imagine a world where then you have a home bakers, guild or association or supply stores where, oh, if you want this bread, might we suggest this blend of these types of flours, these types of yeast and bacteria, right? And then you'll get this bread. I mean, that would be incredible.
Yeah. And then we could also maybe for once and for all, like, figure out the New York bagel thing. And this is a question we're plagued by. Yeah, exactly. But yeah, that sounds like amazing. And there's no reason ultimately that we shouldn't be able to get there.
Yeah, exactly.
It's knowable, but it's a huge amount of work.
Funding.
Yeah. But that is amazing. So I can't wait to check back with you and find out what the updates are. It has been so fun to sit down with you, Erin. Thank you so much for sharing.
Thank you so much for having me.
You need to let us know when you're in New York and come to our studios. We are inside a shipping container in a pizza restaurant in Brooklyn.
I can't think of a better place to be.
Yeah, it's pretty great.
I can't wait to visit.
So we're excited to have you. Thanks again. This has been Dr. Erin McKenny joining us on Heritage Radio Network on tour from On the Rise, the International Symposium on Bread. Our coverage today is brought to you by Charlotte's Got a Lot, and HRN on Tour is supported in part by the Julia Child Foundation for Gastronomy and the Culinary Arts. Heritage Radio Network is a member-supported nonprofit. We appreciate all of your support. Thanks for listening.
Thank you.